And finally, we had the pleasure of hearing from Sturgis Moskos, and he's going to talk about melanoma, another new member of our faculty.
Dr. Murphy, Dr. Moss, thank you very much for inviting me, giving me the opportunity to speak about a very high-speed train that's called metastatic melanoma right now, as changes occur within a few months at a time, and therefore still the field has not settled down. What I would like to try to do is to, first of all, I have been in the advisory board for Amgen, and what I would like to do is I would like to try to move beyond the standard concepts that have been developed over the last couple of years, that, you know, melanoma is B-Raf mutations or N-RAS mutations. They're much more, and we have been hearing it during the session about heterogeneity of tumors, and pretty much I'm going to be discussing the heterogeneity and how do we deal with it at the clinical level. So the outline is very simple. So we will try to define melanoma subtypes in clinical and genetic terms. We'll talk about what is broken, meaning what kind of genetic changes occur, because we all here as an audience believe that cancer is a genetic disease. So we'll talk about what is the impact of any of these changes that we see and how to fix it. So the first two parts are going to be lumped together. Shortly after the pivotal paper in 2002 by Davis et al., talking about the high frequency of B-Raf mutations in metastatic melanoma, the group from UCSF really put together what makes, in 2012, intuitive sense. That is, that there are many different subtypes of melanoma. We have known this for many, many years. They're not only cutaneous melanomas, but there are many others. So putting together environmental factors, and the most frequent of these is sun exposure, and putting together some genetic factors, and that just putting all this together, coming up that melanoma is clinically and genetically a heterogeneous disease. So the group in 2005 described that there are pretty much 4 types of melanoma, and 5 years later they added one more. And basically, this is defined as those melanomas that occur in the skin, but they do not have intense sun exposure. Other melanomas that are in clearly sun-exposed areas of the body. So these are called non-chronic sun-damaged melanomas. And these two groups take up approximately 80% of all the cases of melanoma that we see. And we have these rarer subtypes, such as the acral melanomas, the ones that occur in the palms or the soles. And these have a different genetic profile. And also we have these melanomas that arise in mucosal surfaces, not knowing yet why nature made these melanocytes being present there. And what they nicely put together is that these different subtypes of melanoma do have different genetic mutations, such that the melanomas that are in non-chronically sun-damaged skin have higher incidence of B-Raf mutations, and B-Raf mutations are not very frequent in other of the subtypes of melanoma, and the N-RAS mutations are frequent in the non-chronic sun-damaged and the acral melanomas. Back in 2005, the group overemphasized the prevalence of c-KIT mutations, and that has driven the field for a number of years now. And we routinely test for c-KIT mutations, but probably, I think it was an overstatement back then. And this work contrasts with the work here, where they focused in ocular melanomas, and they found that these types of melanomas are actually sharing an entirely different genetic mutation profile in groups of genes that are associated with G protein signaling. So in parallel with this effort, of course, there is this COSMIC database that has the kitchen sink list of all the mutations that have been observed in melanoma. You know, you're going to be seeing this figure through how they talk. But pretty much most of these mutations do occur in 2 major pathways. So you have the B-Raf mutations and lower frequency mutations in the MEK. And then you have aberrations or mutations in the PI3 kinase pathway. either as loss of PTEN or a very low incidence of mutations in the PA3 kinase by itself, or even a smaller incidence of mutations in the AKT. And then you have mutations in the RAS family of proteins, predominantly in the N type and less frequently in other types. And then you have not so frequent mutations in other types of receptors. I want to emphasize here that these two pathways are very well interconnected, such that if you disturb therapeutically that arm, then that arm appears to be actually compensating or trying to overcome several of the blockade that is introduced, and you will see that later on. Now, we have been talking about mutations, but we should not forget there are also other types of genetic aberrations, such as loss of or gain of copy numbers of genes. And we should not forget that the P16 gene that has been the standard genetic test for developing melanomas. What this group did in melanoma cell lines that were established from patients' tumors is basically to correlate mutation data with copy number data. And there have been 3 different groups. Whenever you have a B-Raf mutation, you seem to also have a higher copy number of the same gene. Same with N-RAS. If you have a mutation in the N-RAS, you tend to have a higher copy number of the N-RAS genes. And you have this group that is neither B-Raf nor N-RAS, as I showed you here, that can have copy number changes predominantly in components of the cell cycle machinery, such as the P16 gene or the CDK4 or the CCND1. It is important to know that the P16 gene, there can be a loss of that irrespective of the type of mutation that you have. And also you can have loss of in other components of the cell cycle machinery. So again, we presume and we operate on the basis that cancer is a genetic disease, and different and every cancer genome has its own way of being dysfunctional. This is a slide from the Meyerson group that they very elegantly presented at ASCO this year, and I find it fascinating because what they're trying to describe in a single slide, mutation frequency manifested as the number of mutations per megabases on the y-axis, and on the x-axis you have different types of cancers. And the point I want to make is that melanoma appears to be the most frequently mutated cancer. And it is interesting that it's even higher than squamous cell lung cancer or lung adenocarcinoma, and then it is much higher compared to childhood malignancies or hormone-dependent cancers like breast cancer or prostate cancer. Now, the interesting part is why is it so frequent mutations? If you look at this part of the slide and you see the types of changes, nucleotide changes that occur, usually those yellow type changes, the C2T translocations, usually are occurring secondary to ultraviolet radiation. So as you can see here, most of these mutations that have been described in melanoma are usually UV signature mutations. And this is very nicely ties an observation that you develop melanoma largely because of your prior sun exposure. I have to mention at this time that the TCGA has been completed for all of these cancers that I put an asterisk on, and still awaiting the official data report for prostate, bladder, and melanoma. So, in the absence of official TCGA data cataloging the mutations in melanoma, what is available? So what is available is this paper where the investigators basically analyzed 121 tumors. This part shows what type of melanomas these were. So green bars are individual tissues. The green ones correspond to cutaneous melanomas. The blue ones correspond to an entity that's called melanoma of unknown primary. And then there are very rare cases of uveal or mucosal melanomas. So the first thing you would like to appreciate is going up here where per each tumor they have described how frequently mutations occur. And I hope that you agree with me that in cases where you can see uveal melanomas or acral melanomas, the mutation frequency in the rest, like the cutaneous melanomas or the melanomas of melanoma primary, is the highest in those types, and it's very, very low in the uveals and the acrals. The other important finding is that melanomas of unknown primary, i.e. melanomas that we do not know where they come from, they look in a mutation profile similar to cutaneous melanomas. So it's very safe to say that a person who has been presenting with a melanoma of unknown primary, probably he or she had a primary melanoma in the skin that for some reason disappeared from the body. Another interesting thing that you see is what we have known for years now, that most of these melanomas do have B-RAF mutations. A subset of these have N-RAS mutations, but you can see that these two mutations are mutually exclusive, with the exception of a very few rare cases here when you have double mutations there. What else do you see? So we didn't need to do a whole exome sequencing as the investigators did in order to get these findings. But what I hope you would appreciate is that the coexistence of other mutations within the B-RAF mutant group or the N-RAS mutant group that suggest a number of things, suggest that within the B-RAF mutant patients or the N-RAS mutant patients, there are subgroups of patients that may actually respond to different therapies. And to the clinicians, this may be a signal to suggest, well, probably there are rational combinations that you can do on the basis of mutation profile. So what kind of rational combinations you may think about? Well, one is to really see that aberrations of the PI3 kinase pathway, either in the form of mutations or functional inactivation or loss of copy number changes, coexist in both the B-RAF and the N-RAS mutant patients. What else do you see? You also see that, again, within the B-RAF or the N-RAS group, there are changes, either mutations or loss of copy number changes in genes associated with cell cycle, like CDKN2A, P16 here, or P14. So the next step, what does all these mutations mean, and how does these affect the patient? Well, what is important to state at this time is that, at least for the highly prevalent mutations that have been studied, is that if you have an oncogenic mutation, it's probably a bad thing for you. And it's a bad thing based on two independent studies, one from the Sydney Melanoma Unit and the other one from the MD Anderson Cancer Center. So what did they do is they took a large number of patients either with a B-RAF mutation and with our prior treatment in the pre-2009 era where no B-RAF inhibitors were available, and then they compared them with the patients who had no B-RAF mutation. And what they saw is that there was significantly worse survival in patients who had B-RAF mutation. And as you can see, at along the same time, there were a number of patients who were actually enrolling into clinical trials for B-RAF inhibitors. And what the B-RAF inhibitors appear to do is they're shifting the curve to actually improving the survival that looks like that is actually slightly better than even the survival of those who do not have the B-RAF mutation. Similar to these data, then you have these data where the investigators, in addition, genotype patients for the N-RAS mutation. And as you can see again, the N-RAS mutant patients did much worse compared to the rest wild type or the B-RAF patients. And this is trying to predict and make prognosis based on a single gene mutation. So what happens if you have 2 or 3 or 4 more genes? We do not know yet, but an early report 2 years ago at ASCO suggested that if you actually have a B-RAF mutation and you have loss of P10, actually this is going to make you live shorter compared to the patients who have B-RAF mutation without loss of P10, or in other words, without aberration of the PI3 kinase pathway. So as we move on and we actually catalog better the mutations that exist, we're going to identify subgroups of patients that will, within the B-RAF or the N-RAS group or any other group, they will have differences in overall prognosis. And why why mutations actually are very bad for you is because we now know that having an oncogenic mutation for the B-RAF is actually an immunosuppressive. So what does it do? Is actually that the B-RAF mutation produces a number of cytokines that basically dictate the adjacent stroma cells, the tumor-associated fibroblasts, to basically upregulate immunosuppressive factors such as PD-1 or PD-L1, such that these factors, when they come in contact with cells, but T cells that are trying to infiltrate the tumor, they either force those immune cells to not proliferate much, not become active, and basically functionally inhibited. So this is how the biopsies in a patient without treatment with B-RAF inhibitor looks like. If those patients actually receive a B-RAF inhibitor, within less than 2 weeks, you will see massive influx of lymphocytes, both effector T cells, the CD8 cytotoxic or helper cells that are infiltrating the tumor. And what happens when patients develop resistance, secondary resistance to B-RAF mutation? The cells are still there, but they have markedly decreased because the tumor has used other pathways. So the story for B-RAF being an immunosuppressive, inducing an immunosuppressive condition has now very well established in the literature. Dr. Thomas does have data in the patients with primary melanomas and N-RAS mutations suggesting that indeed N-RAS mutation is doing the same thing, suppresses the number of tumor-infiltrated immune cells. So are we done? Are we done with, like the TCGA is going to give us all the mutations that the melanomas have? Are we done? Do we have all the information available? Well, the problem with the TCGA is that there's not a well clinico-pathologically annotated database of mutations. So as we're becoming proficient in terms of genetically profiling the tumors, we will see that associating these gene mutations with clinical pathologic data will give us a more complete picture of what does that mean to the patient. So this is a total of 15 patients that have been undergone exome sequencing of approximately 200 genes within UNC. The genes that have been profiled are genes that have been presumably associated with cancer initiation. These have been profiled in Dr. Sharpless lab, and this has been a collaboration among the surgeons and the molecular pathology lab. So this is how data with mutations would look like as, again, we're getting more proficient. Mutations by themselves would not mean anything to us unless we really associate them with, the standard clinical pathologic factors, presence or absence of prior therapies, as well as overall survival. So you can see here that, as we predicted, that we do see mutations. So we do see lots of mutations sometimes, and with black you see the single nucleotide changes. With red, you do see shifts or deletions or other things. So if you look now, if you hypothesize that the number of mutations or the type of mutations is associated with survival, then you would like to see how the number of these mutations is correlating with how long the patients lived. And there are a number of cases here, a number of patients where they have lived for a long period of time, 74 months, 204 months, 71 months, and they had a very high number of mutations. So what I'm trying to say and emphasize here is just by measuring mutations does not really give you the overall big picture. We were able to find the common mutations in these 15 patients. So they were able also to find the less frequent mutations, the N-RAS, the P10, the c-KIT, but also they were also able to identify mutations in genes that have not been previously described, For example, this mutation in the B-Raf, where it is not in the 600 codon, or mutations in proteins involved in the double-strand DNA pathway, like the ATM. So really going after each patient for a particular set of mutations, you may identify genes that were not previously thought that they would be found there. So how are we going to fix that now? If you believe that the FDA-approved therapies are really a barometer of what is important in a cancer, then you have to appreciate that the group has been split between those who believe that non-immunotherapies are effective or immunotherapies are effective. And as you can see, two immunotherapies have been FDA approved, and two non-immunotherapies have been FDA approved so far. So again, it's another evidence that this cancer is not only amenable to chemotherapies or molecular targeted therapies, and the fact that some immunotherapies have been effective tells you something about the biology of this cancer. And in fact, there are equally other types of therapies, both non-immunotherapies and non-immunotherapies, that are coming into surface and they may most likely be approved. So I will focus on these two types of therapies that are basically trying to inhibit the inhibitors of the immune system, as these are becoming more and more prevalent now. So the process of activating a T cell when it comes across with an antigen-presenting cell is very, very complex. It is not only important for the T cell to recognize the peptide that is being produced by the antigen-presenting cell. This has to be seen in the context of a very large number of molecules that activate or inhibit actually that signal. And as you can see, there are lots of them. CTLA-4 is one of them. PD-1 is another. So there are signals that are positive or negative into the activation. And these ones will dictate actually whether the T cell is going to be activated or not. And these group of molecules that are called immune checkpoint molecules are becoming very, very important. We already had one, Ipilimumab, that was approved. In fact, pharmaceutical companies are very much interested, and whatever is a solid arrow indicates how far pharmaceutical companies have developed antibodies against these proteins. And with open arrows indicates targets that are currently in preclinical development. But it's not only important whether there are co-stimulatory or co-inhibitory signals, it's also important where do these get activated. Because if a T cell is occurring in the lymph nodes, then what is more important is to have a CTLA-4 there. But if the cell is found in the tumor tissue, then other proteins are more important. So why is this important? It has to do with the side effects. That can explain very well why CTLA-4 blocking antibodies have more significant side effects, autoimmunities, compared to the PD-1 antibodies that have a better toxicity profile. This is the pivotal trial that led to the FDA approval of Ipilimumab. Ipilimumab was given alone or in combination with the GP100 vaccine, and whoever got Ipilimumab lived longer compared to those who received only the GP100 vaccine. As you can see, the effect is only applicable to only a very small group of patients, 20% of them, and it takes time to really see the differences. I don't have time to go into details, but this is exact same Kaplan-Meier curve that you get with high-dose bolus interleukin-2. Unfortunately, there were not any biomarkers that can help you predict the response to Ipilimumab. So right now, this drug is available to everybody who has melanoma, irrespective of the B-Raf mutation. However, antibodies against the PD-1, either developed by BMS or developed by Merck, have shown that irrespective of the doses that have been administered, actually the response rates have been striking in phase 1 trials. Between 19 and 41, depending on the cohort that was studied for. And as I explained earlier, because PD-1 is overexpressed only at the tumor site where chronic inflammation occurs, the toxicity profile is very minimal there. And in fact, we think that there is a predictive biomarker that may predict the response to a PD-1 antibody therapy. And this is the expression of PD-L1 in tumor tissues. So if your tissue expresses PD-L1, which is a marker of chronic inflammation, probably you have high likelihood to respond to a PD-L1 antibody. This is the pivotal trial that led to the FDA approval of vemurafenib. This is the update analysis that Paul Chapman presented at ASCO this year. So this is the updated overall survival curves after the drug, it was allowed to be given to all patients. So vemurafenib holds an overall survival benefit after crossover was allowed compared to the carbazine. We're seeing the exactly same thing with dabrafenib, which is another B-Raf inhibitor developed by GlaxoSmithKline. So irrespective of the B-Raf inhibitor you use, actually you get the same benefit compared to the carbazine. This is a predictive, so we were asking why the response rate to B-Raf inhibitors is actually only 15%. So when we were part of the phase 2 trial of single-agent vemurafenib and we collected tumor biopsies before, we asked the question, what can predict a response to vemurafenib? And what turns out to be is that if your tumor does not express immunohistochemically P10, okay, that will make you to be a non-responder. So what we think is going on is those people who have who receive B-Raf inhibitor and they respond, probably this is the group of patients where the signal is dominantly coming through the RAS, MEK, and ERK pathway. Whereas those patients who are not responders, probably because they do not express much of P10, this arm is more active, and therefore the signal is not as dominant to really lead to a clinical response. Yes. MEK inhibitors have become more coming into surface right now, and right now GSK is getting approval, is trying to get approval of the MEK inhibitor. This is a trial that showed that the MEK inhibitor in patients with B-Raf mutant patients is doing better than chemotherapy. We're beginning to see effect of MEK inhibitors in N-RAS mutant disease. And I'm going to skip the, and I want to show you this, is that if you now combine the B-Raf plus the MEK inhibitor, there is early evidence that the progression-free survival with the combination is better, and it's reaching a 9.2 progression-free survival benefit compared to single B-Raf alone. I want to close with the idea of combining immunotherapies with molecular targeted therapies is a very important one. There are only a few trials that are currently in progress, and the early data suggest actually that the combination is very, very toxic, and therefore adjustments need to be made. Thank you very much.